Today we start the third part of the course. As I told you in the beginning, the course is composed of 4 parts. The third part is about the construction of the research project. Hopefully, what this part here would like to convey to you is the idea of how a research project is conceived of, then elaborated, carried out, and its results get interpreted and converted into scientific facts. So I'll have you involved in several exercises during this series of lessons. So the construction of the research project can be subdivided, and this is my personal subdivision of the research project into 4 levels. In fact, there would be 5 levels. One is the execution, the material execution of the experiments, but we have talked about that a lot already, so I won't go into it. But the 4 levels I want to talk about are: first, the question; second, the experimental approach, or the observational approach. It is an observational study. The design of the experiment or of the observation, then the execution of the project, and finally the interpretation of the results. Now, as I already said, this is my personal subdivision. You won't find it elsewhere. Other people might describe a research project in a different way. And what's important is that for didactical purposes, we'll talk about these planes, these levels, separately. We have to. But you should remember, and I'll remark it occasionally, that the levels are somehow overlap one another, so that you never finish to ask a question before going to the lower levels. You just keep the question going. And same here, you may have come up with an experimental approach, but then as you design the experiments you realize that the approach must be changed, modified, and so on. So the levels are somehow have a gray boundary among them. But so let's start with the question. First of all, the question must be a scientific question. That is, a question that can be approached with a scientific method. We have already talked about what part of the human knowledge are not scientific, which doesn't mean they are not valid. They are just not approachable with the scientific method. And so scientific questions must be approachable with the scientific method. But that's not all. One thing that is very important about scientific questions is that you should focus the question. And I'll show you what I mean by focusing. I mean that if you clearly define the problem, that is, if you ask the proper question, then it becomes a lot easier to find a way to solve the problem itself. The first example I want to show you in order to exemplify this concept is very well known to you, and it is once again Yamanaka's paper. So we might imagine that Yamanaka started from a very general and in fact a generic question. Is it possible to convert somatic cells into stem cells? Well, nice question, of course. But it is very generic. It doesn't suggest any specific approach. So 99% of people being asked this question would answer, perhaps, I don't know, that would be it. But Yamanaka went beyond this point and tried to focus the question. And so we might, again, imagine, we don't know exactly what was the thinking, Yamanaka's thinking, but we might imagine that the next form, the next shape this question took in his mind was, can transcription factors induce this conversion? Why did he think of transcription factors? Because if you remember the background to his project, to his study that I told you about several lessons ago, he knew already that at least in one case, fibroblasts could be converted into skeletal muscle cells by expressing a single transcription factor called MyLG. And there were other pieces of evidence in the literature that suggested that transcription factors might possess the ability to induce this conversion. But then again, transcription factors are many. They are not necessarily the sole tools that you might need to induce the conversion. So again, this question is not focused enough here. An even better question is, can specific transcription factors induce the conversion? So the transition from this to this question is the word specific, meaning let's reduce the number of factors we are going to consider. Not all of them, there are hundreds. We need to consider a few of them so that the question can be handled with experimental procedures. And so in fact, Yamanaka went through a long exercise of selecting transcription factors based on previous knowledge, based on intuition perhaps, based on the fact that they had been previously shown to be either involved in stemness or expressed specifically or exclusively in stem cells. So eventually Yamanaka came up with 24 transcription factors that he selected, and that made the approach he chose feasible. So the question in this form is focused enough that it suggests an approach. It suggests that if you are good enough to select the transcription factors that might be involved in the process you are going to investigate, then you can fairly easily devise a method to investigate those transcription factors. And in fact, we know what Yamanaka did. He engineered cells in a way that they would be selectable if converted into stem cells. So we can suppose that Yamanaka started from a generic question such as this, and then by a process of refinement improved the question up to the point when the question itself suggested a possible way to approach to find a solution to the problem. So let's see another instance of this kind. This is basically a random paper in the literature that I'm showing you. In fact, it is a paper that I should have shown you in the previous lesson, but I didn't because that lesson was delivered by my colleague who chose another paper. But never mind, because we can summarize it. I'm saying that this is basically a random paper, meaning that in almost any paper you could do the same exercise that I'm going to do, we have done here and I'm going to do in the next one. That is, finding what probably was in the mind of the experimenters, of the scientists, as a generic question, and refining and focusing it until it was ripe, it was mature, it was ready to suggest an approach. So, in this case, the original question was, is there a microbiological cause for Merkel cell carcinoma? What is Merkel cell carcinoma? Merkel cell carcinoma is a very rare tumor that almost exclusively arises in immunodeficient subjects. And so many people studying these tumors thought that perhaps it was among the few tumors, the few human tumors, that are caused, or I should say co-caused, by an infectious agent. Why did they think of it? Because if you think that the subjects that develop the tumor are immune deficient, it is very possible that they are prone to being infected by a potential tumorigenic infectious agent and then develop a tumor much better, much more frequently than the general population, the general immunocompetent population. So, the generic question was clear in their mind. Is there microbiological cause for Merkel cell carcinoma? Now, where did they start from? What do they know already when they started with this question? Well, they knew that several human cancers are caused by, co-caused by infectious agents. I'm underlining co-caused because no infectious agent alone is capable of inducing a human tumor. That's very important, because if it were, we would have an epidemic of tumor caused by an infectious agent. That's not the case. They also knew that infectious agents can be either viruses or bacteria, or at times even parasites, these non-unicellular organisms. And that in the case they are viruses or bacteria, they are not always necessarily found as whole agents in the tumor. Sometimes all that's left of them is an oncogene. That is, the specific part of that infectious agent that is necessary to cause the tumor, or to increase the probability of developing the tumor. And so they were facing a very serious problem here, because they thought there might be an infectious agent. They knew nothing about this potential infectious agent. They didn't even know whether it was there or not. They knew of none of its characteristics. They had no idea what to look for, and they had no idea whether it was there at all, at least as a whole intact infectious agent. So, now I'm asking you, what would you look for in these tumors if you were looking for an infectious agent that caused them? I have already given you a clue. You paid attention to what I said.
The presence of microbiological DNA or RNA or proteins.
Excellent. Other ideas?
Look if the host have any symptom of infection like fever and... No.
That's not a good approach because of something that you probably don't know or you don't have present in your mind. There is a very, very long lapse, like tens of years, between an infection and the eventual development of a tumor. So there is no such a thing as acute symptoms of an infection at the time when the tumor develops.
I don't know, maybe because it can be later years of the infection, maybe there will not be the biological remains, but you said that maybe un agente cancerogeno,
I said an oncogene.
an oncogene, yeah.
What is an oncogene?
I don't know.
You don't know what is an oncogene?
It's something that makes cancer more likely to happen.
Yes.
Okay.
It's basically a gene which can be endogenous or exogenous, depending on the circumstances, that contributes to carcinogenesis.
So maybe look for the oncogenes that we know.
And so, well, no, I'll stop there.
Okay, not.
But but if you're looking for an oncogene, then you're following Valeria's suggestion.
Yeah.
You're looking for genetic material, basically. So, and so through a process of progressive refinement, probably the authors came to this idea. Can we find genetic traces of a causal agent? As I said, at times the causal agent is not there. Say it's a virus. You can look at the microscope, even the electron microscope, at the tumor, and you won't find the virus. But in the instances of infectious tumors that we know of, there are always at least remnants of the genetic material of the culprit, of the infectious agent, usually, but not always, integrated into the genome of the host, independent of the fact that the exogenous material is integrated. Nonetheless, we expect to find some genetic traces of the infectious agent, because in all the instances we know of, the oncogene must remain in the tumor cells in order for the cells to remain tumorigenic. If they lose it, they either die or stop growing, or anyway do no longer display the tumor phenotype. So let's go back to Valeria and ask Valeria, okay, we are looking for genetic material. We are looking, proteins is more difficult. It's not impossible, but it's more difficult. We are looking for oncogenes, possibly, likely, but we don't know what oncogenes we're looking for. How would we do that? We are looking for a sequence from an exogenous agent that is in the tumor, that is present in the tumor cells. And Samuele,
Maybe comparative genomics.
Go ahead. Yeah. Valeria agrees? Okay. So how would you do that? Practically. What are you going to do tomorrow?
Sequencing the
Yeah.
cell of my patient and sequencing the
And
tumor's cell
Yeah.
and compare.
It's even simpler than that. You can sequence the tumor cells from the Merkel cell carcinomas and then compare them with databases containing
yeah.
the whole human genome. And that's what the authors did. And by doing that, I don't have the slide here, but nevertheless what they did was they sequenced the genomes of these cells, then they eliminated more than 99% of the sequences because they simply aligned with the human genome. And so they eliminated them. They were left with a few reads, and among them they found one that was similar to a simian polyomavirus. Polyomaviruses are tumor viruses for many animals, But until this paper, no polyomyelitis was found to be tumorigenic for humans. This is the one and only exception we know at the moment. And it is tumorigenic only for immunodeficient humans. But nonetheless, by subtracting, as you suggested, the human sequences, they were left with foreign ones, or at least unknown ones, and among the few unknown ones, they found indeed something that resembled, indeed was an oncogene. And from that, they reconstructed the whole genome of the virus, which was indeed present entirely in the tumor cells. Not the virus itself, but the genome was there. Now, you may not have realized it, but together, the three of you already generated a research project. I gave you a question, and you devised a way to approach the question. That's the nucleus of a research project. Now we're going to refine this process, but you have done it already. So it is not that difficult, it just takes thinking, reasoning, and knowing, of course, a background to your question. All right, so let's see a different example. Oh, this comes from a personal experience. When I was young, I was among the first scientists to work with PCR. It had been already, it had been just invented when I came to my first laboratory in the United States, and my boss put me to work on PCR to find a specific translocation present in human lymphomas. And at the time, the PCR was so new that there were no PCR machines. So there was nothing cycling through temperatures in order to do the reaction. And what I was doing was, I usually did these experiments on Saturdays because then I could sequester all the water baths in the laboratory, put them on my bench, and have them set at different temperatures, and then I was cycling my tubes from one to the next many times until the PCR was done. More important, Taq polymerase, the heat-resistant polymerase, had not been isolated, it did not exist yet. So at the time, people were using Klenau polymerase, which is DNA, E. coli, the E. coli DNA polymerase, like the fragment of it, which is thermal sensitive. So at each cycle, when I put DNA at 95 degrees, it got destroyed. So I had to open the tubes, add more polymerase, close the tubes and cycle them again, and then reopen and then add, and then close and cycle them again. It was very tedious, it lasted several hours. I couldn't leave my bench, I couldn't go to the bathroom, I couldn't do anything. So, how is a PCR done? I think you know. You need a template DNA, you need 2 primers, you need nucleotide triphosphates, which are the building blocks of DNA, and the reaction, of course, you need a polymerase, and the reaction takes place in a 1x buffer at the time with this polymerase at pH 8.3. What is a 1x buffer? I don't think you know. 1x, do you know? No, okay. It's basically laboratory jargon. 1x means the concentration of use, the concentration that is really present in the tubes that you're working with. Now, oftentimes in the laboratory, you don't prepare solutions at the working concentration, because when you work with those, you have to add other ingredients. And so the volume increases, and if you start from the right concentration, it gets diluted. And so finally, you don't have anymore the right concentration. So you prepare concentrated solutions. In this case, I had a 10x buffer, which then got diluted by the other reagents, and finally added some water to bring the buffer to the right 1x concentration. Good. One day, I had a particularly long experiment, and I finished it. I froze the products, ready to analyze them the next Monday. And then I sat down at my table, and like a good scientist, I recorded everything I had done. And while writing down the procedure, I realized that I didn't remember if I had diluted the buffer. And as strongly, as much as I could concentrate on the issue, I couldn't remember whether I had or not diluted the buffer. And damn it, I had only two choices. Either repeat the experiment right away, or going through 2 days of analysis to determine whether the PCR had worked or not. And if it had not, I had to repeat it anyway. So neither solution was appealing to me. I was really banging my head on the wall, but then realized that I had to do something. I had to find a way to determine what I had done. So the question I had in mind initially was, of course, generic. Was the reaction successful or not? Did it work or not? Now, that was really the question that I needed to answer. But this way, this kind of question does not suggest an approach. So, the question to you now is, how can I refine this question? Why might have the reaction possibly not worked? Just told you.
Because the concentration
Because the
of
concentration of the
was
buffer
not
was
right.
wrong. Good.
So easy, not, but was the concentration of the buffer?
I didn't.
Yes, it's the same question.
But if you focus on the
Ah.
buffer,
No. I was thinking for the volume. And
no, the volume was okay.
Okay.
What's the purpose of a buffer? Remember first year chemistry.
The pH.
Keeping the pH put. Right, so
Is there a variation of pH after the reaction?
minimal, almost negligible, I would say. However, keep thinking on these lines. If I had a 10x buffer, what would you expect?
Compare that they have the same pH. So compare.
No, actually, a 10x buffer has basically the same pH as a 1x. Not exactly, but undetectably different with conventional units.
If we add like a strong base or a strong acid, the more concentrated buffer will soffre di meno del...
Yes, exactly. So the refined question was, is the pH 8.3 buffer 1x or 10x? Because that was the potential problem. So first of all, focus on the actual agent, the actual problem, and then find a way to approach the question. But this question already suggests a solution, that is what you've just said. A 10x buffer is 10x better at keeping the pH put. So, what did I do then? You tell me. You already suggested a procedure.
It's a titillation.
Titration, yes. Titolazione in Italiano.
Giusto.
Com'è in inglese?
Titration.
Titration.
Yes. So, a practical procedure on the bench. What will you do?
We are going to need instrument that measures the pH and
Yes,
measure
that's
it
easy.
while we keep adding either a strong base or acid.
Yes, correct. So, the question might be, how much HCl, hydrochloric acid, do I need to add to the solution in order to shift the pH to something lower, say 5? I know I'm starting from 8.3, so how much do I need to add to go to a pH 5? And so, how many tubes do I have in front of me?
A lot.
Well, all of my PCR tubes are the same, so I just pick one. That's my test tube. But actually I'm doing an experiment. What do I need to have in an experiment?
Control
Controls. What controls?
the real buffer before we put it, like the one that you have stored in the lab.
Well, I didn't keep any
The 1 pair or the 10 pair?
the 1x or 10x and 10x.
Ah, oh, okay.
So, what I did in fact was take an aliquot, small amount of my PCR, one of my PCR reactions, then add to it hydrochloric acid and compare it with 1x and 10x buffers. So it turned out that it was 10x, unfortunately.
So you
But at
know.
least I needed to repeat the experiment, but I didn't go through 2 days of laborious detection of results that weren't there. So that I gained something from this. And so we've had another example of focusing a question in order to find an answer. And we could go on and on, but I think you have got the point. You don't just start doing experiments once you have a question. You get questions every day. You have to really think about your question. Try to refine it as far as possible. Maybe you need to read about it. You need to read the background to that question. What is already known. That should give you suggestions, clues as to what to do. Once you feel you have refined your question enough, then look at it. It will suggest you something. So take your time, work on your question, then continue. But then, remember, the schematic we talked about in the first day. The schematic according to which science is done in this way, we have previous knowledge, we have a scientific question, from which, to answer which, we hypothesize something, then we put the hypothesis to test, and if it is correct, if it is sustained by the test, then we draw conclusions and establish scientific fact. If it isn't, we go back and come up with another hypothesis. All right, but then I have another question for you. We have already considered this point, and to ask this question, I'll start from this paper, Philosophies of Funding. It was a commentary that is not a research paper. It was just a theoretical elaboration by philosophers of science, who started their short paper by saying philosophy is often regarded as abstract and irrelevant to practical concerns, which is much what we think about philosophy. But they showed in this paper that philosophy is indeed very important when it comes to funding science, because You have to philosophically define what science is. Otherwise, you don't know what to find. And so, we know the schematic I've just shown you. And we go back to Yamanaka's paper and ask, what was Yamanaka's hypothesis?
Some factor, transcription factor, can revert the somatic cell to
Convert,
stem
yes.
cell.
Yeah, but that's a rather vague hypothesis. It is a hypothesis.
These 24 factor can, some of these.
That's a better hypothesis, yes. But in fact, what Yamanaka was doing was a screening. We haven't yet talked about screenings. We'll do after this section here because of practicalities with my colleague. We were somehow modifying the order of lessons in order for her to participate. Anyway, Yamanaka had a number of potential of transcription factor potential involved in the conversion, but he really didn't know which, how many, or if any of them would induce the conversion. What he did was doing a screen. He went through all of them and worked out which one of them was in fact capable of inducing the conversion. So he didn't have a strong hypothesis. He didn't think, I know that this one and these ones are the transcription factors I need in order to obtain the conversion. That would have been a proper hypothesis. His hypothesis was very weak, vague. There might be transcription factors, maybe they are among these 24. So in fact, he didn't have a hypothesis. And yet we don't think Yamanaka's paper was not scientific. The thing is that the schematic I've shown you does not apply to all of science, as the authors were saying in this paper. Science is more than testing hypotheses. And if you don't realize that, you underfund or you do not fund important parts of science. And so I want to show you very bright examples of science, stellar science, historical science, landmark science that does not conform to the hypothesis testing schematic. So when Darwin went around the world, he wasn't thinking of selection. That he elaborated his theory later. He was just collecting data. Collecting data is a scientific endeavor, is a scientific activity. It may sound trivial, uninteresting, intellectually not stimulating, but data are essential to do science. You don't do science without data. When van Leeuwenhoek invented the first microscope, he didn't know, he had no idea that he would discover a microscopic world. Nonetheless, he invented an instrument that made infinitely more discoveries than any specific hypothesis that has ever been made. So, building instruments, inventing instruments is a scientific endeavor. When Galileo pointed his telescope to the sky, he didn't know he would see the phases of Venus, the satellites of Jupiter, the sunspots. He just wanted to know what was there. The lunar craters. When Newton devised this gravitational theory, he relied heavily on very careful observations by the Royal Astronomer John Flamsteed, to whom he commissioned specific observations. He needs the data, he needs the measurements in order to devise a quantitative law. so measuring is often considered an activity for technicians, but it is very, very basic to science. And so science is much more, is many more things than just testing hypotheses. That's a very, very important part of science, but by all means not all of science. And so other realms of science besides hypothesis-driven research, which is another way of saying hypothesis testing, so it's a research driven by hypothesis. But other than this, oops, I haven't translated this. This is question-driven research, that is exploration. I messed it up. No. Measuring is also science. Inventing instruments, constructing, building instruments is also science. Remember this if people tell you that what you're doing is not science. Highlights. The question is the starting point of the scientific project. Again, take your time to develop a good question. Scientific questions or objectives include hypothesis testing, exploration, development methods or tools, and measurements. All of this is science. Comments? Okay. Then we go to the second level. Second level is the experimental approach. Here it is. So say we have refined our question, we have clear in mind what the question is, and now we need to find a way to approach the question. That's the experimental approach. Now, what is it? That's a kind of broad brush painting of what you're going to do. Guidelines as to what to do, not refined, not defined in the details, just a general picture of what the approach is going to be. I always make a parallel with the work of an architect. When an architect imagines a building, he doesn't start from the electric system or the elevator tube. He starts from a drawing of the whole building. Then he designs the details. And so does a scientist. First of all, he imagines the approach, the whole study as a whole. And then works out the details in terms of designing specific experiments. But definitely, these levels comes first. You have to have a general drawing of the building before making projects of the details, right? Otherwise, a detail by itself, you don't know where, it doesn't fit anywhere. Good. So, how do you work out an experimental approach? Well, there are many ways. In my mind, the best way is by discussing it in the laboratory with all the people involved in the prospective study, and if at all possible, even people that will not be involved in the study, but that are just interested, just because they are in the laboratory, elaborate the same ideas, work on the same subjects, so they might be interested in participating. And these are really one of the best times in the laboratory. When you are approaching a problem when you're approaching a question, the problems that you will certainly encounter while doing the study are not there. You just think of ideas. You just think of ways to approach the problem, and you see the solutions already at a distance. So it's really a sort of an emotional discussion in a way. The faces of these people really tell what the atmosphere should be and often is. Indeed, everybody's optimistic because you see that at the end of the road, the answer that you're seeking is there. There is a way in which these discussions should take place. We talk about when we discuss the ethics of research, but I will anticipate that everybody should contribute to the discussion. Even the young people who might be inexperienced and possibly not very knowledgeable about the subject, they might contribute ideas which are valuable just because they come from people that haven't gone many, many times through the same lines of thinking and keep thinking the same way. They might give you completely different ideas, completely different suggestions that might be very valuable in designing an approach. Of course, the older people in the lab that are more experienced and knowledgeable will try to select these suggestions, and they will also try to refine it, because they will probably be gross in the beginning, because they come from inexperienced people. But they should be open and listen to the young people. If they don't listen to them, they don't get the point, they don't get the new ideas. So everybody should take his own place in the discussion. But then again, what happens in these discussions? Well, exactly what's happened a few minutes ago when you were trying to develop a way to approach the question of infectious agents in the Merkel cell carcinoma. She said something, you refined it, you had added something, and so on. So it's a process of progressive accumulation, refinement of ideas. Oftentimes it involves discarding ideas that initially look good, but then we find that they are not good enough, or that there are better ones, or they won't work at all, so we discard them right away, etc., etc., etc. It's an iterative process. It also takes quite some time. Sometimes you discuss projects and you don't even know that you're doing it. You just discuss ideas, and then they stay there for a while, sometimes for a long while, and then eventually they become a project because something new comes up, and you find that the ideas you have discussed in theory without even thinking of applying them now make sense, and now you can discover something that you haven't even imagined before. So, once again, the approach is a broad brush picture of what you're going to do during your experimental or observational study. And you can subdivide the approaches into two very large classes. One is this, the other one is this. In the first class you find hypothesis testing, which we have also called hypothesis-driven research, which you might also call aimed approach. You know exactly what you want to reach. You already suppose that your hypothesis is correct, so you want to reach a confirmation, support for your hypothesis. On the other hand, you might take a blanket approach. I don't know what's there, but I want to know. In order to take a blanket approach, I'll need some sort of brute force. In fact, these sometimes are called brute force approach. Sequencing the whole genome to find just a few foreign sequences is a brute force approach. You don't have to select, to isolate, to guess what they are. You just sequence the whole thing and find what you're looking for. It is also called unbiased, without preconceptions. And that's interesting because I think we already mentioned that hypotheses, as good as fundamental as they are for science, are preconceptions. At the very moment in which you come up with a hypothesis, you have a preconception. You already have an expectation as to what the next experiments will yield. Instead, in these kinds of approach, you don't have biases, you don't have preconceptions. You just look, you just measure, you just find out what's there in the field you are exploring. They are also sometimes called in a derogatory way fishing expedition. You just throw a net and you'll pull out something. That's considered, it's derogatory because people who use this expression here imply that you are conceptually weak. You cannot think of a good hypothesis, so you just throw a net and fish out something. Well, sometimes that's when we talk about screenings, we'll see that sometimes that's the only way to approach a problem. So anyway, the two classes, hypothesis testing and the blanket approach, the unbiased approach. All right, so I want to reinforce the idea that science is by all means not confined to this section here. By telling you a story which sounds like a fairy tale. Here is Henrietta Swan-Leith. She was a very talented woman at the end of the 19th century in America, in Boston. She, being a woman, she wasn't entitled to a scientific education. She was not even supposed to have a job. So she worked for free in the Harvard Observatory, where her boss was employing her. In fact, it's not even correct to say she was employed because she didn't get paid. She was using her as a computer. At the time, there were no computers in the sense that we intend today. So computers were human beings, most often women, because they are thought to be more accurate and they cost less. So complex calculations were done in this way. You had a series of people aligned on a long table, and each one of them was doing one or a few mathematical operations and passed the result to the next one, and so on. And there was often somebody going back behind them, looking at what they were doing to make sure they weren't making mistakes. That was computing at the time. So Henrietta Swan-Leith was a computer, and they found that she was really good at math. And so eventually she got even a bit of a pay, and even later she showed that she was so good that she was put at a different task. She was tasked with studying variable stars. What are variable stars? Are stars whose luminosity changes with time. Now there are two main kinds of variable stars. The ones that vary casually and the ones that vary very, very regularly. Those ones are called Cepheids because the first ones were identified in the constellation of Cepheus. So what Henrietta was doing was a really horrible, painstaking work. She took photographic films of exposures of the same section of the sky day after day, and she compared the brightness of every star one by one until she found one that was whose brightness varied in time. Then, when she had found one such star, she had to measure the amount of variation and see whether there was a constant timing in these variations or not. And she was interested in the Cepheids, in the stars that vary their brightness in a constant way. And she spent several years, and eventually she cataloged nearly 2,000 of those cells. You can imagine how difficult and how painstaking this work was. But she was so good that she didn't stop there. She noticed that there seemed to be a correlation between the absolute brightness of those stars and the length of their period. The brighter the star, the longer the period. Now the problem is that the brightness of a star seen from Earth depends on the distance. So, she had a very good idea. She thought, I'm going to look at Cepheids that are all in the same place, and so presumably at approximately the same distance from Earth. She looked at Cepheids that were in a Magellan Cloud. What is a Magellan Cloud? It's a sister galaxy to our own galaxy. There are two, a major and a minor Magellan Cloud. I don't remember which one of them she took. But anyway, she supposed correctly that all the stars in the Magellan Cloud were approximately at the same distance from Earth. So she could take relative brightness as absolute, because they were all at the same distance. And yet she found, this is an example of Cepheid, and indeed she found that there was a correlation between brightness and period. And since there was this correlation, she now had a way to determine the distance of stars. When she saw a Cepheid, by measuring her timing, she could determine what was its absolute brightness, not its relative brightness. And so this method is basically what every method we ever had to measure distance from stars. Even the more modern method relies on the Laplace method in order to determine these distances. So this is an example of how a very tedious measuring work became a landmark discovery in astronomy. Now, let's suppose we have a broad picture of what we want to do. We suppose that a certain approach we have elaborated will take us to the answer we're seeking. Well, before undergoing a long, labor-consuming, costly, expensive study in the laboratory or in the field, we want to be reassured that our chances of success are at least relatively high. So we want to evaluate the chances of success in advance. We want to evaluate how likely it is that eventually our approach will lead us to the answers we long for. And so there are many ways to do this, and scientists probably use a combination of all of them. One thing is just to run the experiments in your head, pretty much like this jumper is doing. You look at her expression. She's rehearsing in her mind the movements she's going to do in a minute. She's concentrating. She's trying and figuring out what the problem might be, what muscles she needs to involve the most, what movements she needs to make, and so on. So she is really running in her mind her actions from here. Her action, the action she's going to take in a minute. And the scientist oftentimes does the same. He's had an idea about how to proceed, and then he tries to figure what he would do at the bench, what problems he might encounter that he hasn't thought about yet, what reagents are missing, what caveats are there in his reasoning, what possibilities, what traps are in his reasoning, and so on. A very well-known example of thought experiment never done in practice, only in thinking, comes from Einstein. He said this has been the brightest idea in his life when he realized that if you are closed in a box, so in an elevator cabin, without windows, you have no opportunity to look outside, you would experience the same thing whether you are in the outer space, there's no gravity, or you are free falling, and you're going to crash at the ground eventually. But while you're falling, you free exactly as if you were in the outer space. You will feel no gravity. And so he realized, what kind of force is this that disappears when I'm falling? It shouldn't. If it were a real force, it shouldn't disappear. And so he realized that gravity was not a force. That's where general relativity started from. So one way to try to anticipate the eventual outcome of your project is simply thinking it through carefully, many times, thinking and thinking and thinking of it until it's very clear in your mind and you feel pretty sure that you haven't forgotten any important points, that you haven't fallen into any traps, that you haven't misinterpreted previous knowledge, etc. Another way to try to estimate your chances of success is doing quick and dirty experiments. Quick and dirty experiments are so-called, let them passion this. Per favore abbassate la voce. Grazie.
Avete ripetuto Andrea?
Sì, io. Non troppo.
So quick and dirty experiments are not as bad as depicted here. In fact, they are experiments that skip a few preliminary results. In fact, they should come later, before you should, in an actual study, you should establish previous facts and then eventually run this experiment. But this experiment is crucial. If it works, your hypothesis is probably good. If it doesn't, your hypothesis is probably not. So you want to do it first, even though you don't have all the bases necessary to do it. Because you want to know already if your approach is viable. So you do an experiment without knowing everything that you should in order to do a good experiment. Perhaps you don't have the proper controls at hand, never mind. Try to establish some confidence in your hypothesis, in your approach. And in fact, Yamanaka's experiment was just such an experiment. We know from an interview of his that this was not meant to be a real experiment. It was just a setup. It was just a first try to see whether there was any good in his approach. But it so happened that the dirty experiment worked right away, and so he got his results immediately. Sometimes one gets lucky. Most times. Okay. So, another possibility which we will spend some time, and it's a possibility that comes into play when you have to make quantitative estimates. In these cases, you don't have a yes or no answer. My hypothesis is correct or no. I need to know in advance how much it is, how much it weighs, how much of my variables I'll measure in the end. It's not black or white. It is all about how much. Then you are in need of a quantitative estimate. And doing quantitative estimates is a bit difficult. It's very rewarding. So I'll show you one way to do quantitative estimates. And then I'll ask you to do such estimates as an exercise in this lesson and in the next one for sure. So one way to do these estimates is to take the Fermi approach, where Fermi is, of course, Enrico Fermi, the famous physicist. He was a genius. He went to university at 17, and he already knew all about physics. It is said that when he had to take an exam, the day before, he looked at the program and said, no, yes, I know this, I know this, I don't know this, okay, and went to the exam. And so, later on in his career, he was known as the Pope, with reference to infallibility. And he was a very rare physicist. He was great at theorizing, at experimenting. So physicists usually are either theoretical or experimental. He was both. And one of his habits was to do calculations on everything. And his approach is so typical that it is called nowadays Fermi problem, or the Fermi approach to problems. And physicists are taught how to do quantitative evaluations. It is very important in physics because oftentimes you are in a situation in which if you want to really have an answer to your quantitative question, you have to go through very lengthy and very expensive experimentation or very lengthy calculations, like months of the computer. So you want to have an estimate first before you go through all this. Biologists think they don't need it, but I'll show you that they do. And so I'll try to show you how you do these kind of calculations. So Fermi had this habit, as I mentioned, of always doing calculations. During the experiment with the first atomic bomb, he was in a bunker together with other scientists and soldiers, of course, far away from the point of the explosion. And they had a lot of instruments in order to calculate the force of the explosion. And when the bomb exploded, he had a few bits of paper in his hand. He let them go and looked at where they fall. And he said, this is I don't remember, it was, I think, a 1.8 kiloton explosion. And then the other scientists went through 2 hours of calculations, came to a very close conclusion. The way he did it was to use knowledge that he had in his mind or that was very readily available. nowadays, if you take his approach, nearly every knowledge is readily available. You just ask the computer, you ask Google, whatever you need is there. So it is immensely facilitated. Let's see what a typical Fermi problem is. Oh, by the way, yeah, to solve the Fermi problems, one exploits, as I just said, known or easily acquirable information, often using approximate values. If you have an information, a quantitative information in your head, probably it's not very accurate, you just have an idea. For example, if I ask you, what year was Napoleon born? You don't know. I don't remember. But if I ask you, in which century was Napoleon born?
1700.
Pure, avrei detto anche io 1700.
Inizio 800 c'era lui, dopo la presa della Bastiglia le cose, arriva Napoleone, non lo so, più o meno fine 700.
Exactly. What has Samuel done?
Approximately.
She had some
Previous knowledge.
dates in his mind and tried to put Napoleon after this and before that.
Yeah.
Right. That's the way to reason. Access some relevant knowledge and use it to give an estimate of the quantity you are seeking. Alright, so here is a question. Oh, questo ho deciso di lasciarlo in italiano perché è un esercizio difficile da imparare, quindi non vi tedio con l'inglese. Domanda alla Fermi: quanti accordatori di pianoforte ci sono a Chicago?
Tanti? Quanti abitanti ci sono?
Impossibile, no? A Chicago perché Fermi ha vissuto a lungo a Chicago e infatti in effetti ha costruito lì la prima primavera. Sembra una domanda impossibile, bisogna consultare il librone. Ecco, ai tempi di Fermi, appunto, Google non c'era. Se vi serviva un'informazione quantitativa, dovevate andare a libri di riferimento, dovevate andare in biblioteca e non è detto che la trovaste, anche se c'era un libro che conteneva quell'informazione, dovevate scoprire quale e poi cercare nel libro dove stava quell'informazione, era veramente duro. Per cui l'approccio di Fermi era fondamentale realmente. Se lo applicate oggi, come vi ho detto, le possibilità sono moltiplicate. Bene, proviamo a rispondere a questa domanda. Questo ve lo faccio vedere io come sindaco. Alle prossime domande risponderete voi. A Chicago vivono 9 milioni di persone. Diciamo che in media ci sono 2 persone per famiglia. Apparentemente ci sono molti singoli a Chicago, non importa, stiamo approssimando. Stimiamo che ci sia un pianoforte in una famiglia su 20. Sarà più o meno così, se pensate nel vostro, tra i vostri amici, parenti, quanti hanno un pianoforte, non molti. Quindi 1 su 20 sembra raggiungibile. Un pianoforte viene tipicamente accordato una volta all'anno. Se non siete dei concertisti, una volta all'anno va benissimo. Quando accendete i musiconi, il pianoforte si scorda e poi si va a un'accordatura richiede circa 2 ore, inclusi i trasferimenti dell'accordatura. Ai tempi di Fermi c'era poco traffico a Chicago, oggi richiederebbe di più, ma diciamo 2 ore. Un accordatore lavora 8 ore al giorno, 5 giorni a settimana, 50 settimane all'anno. Bene, questo è tutto. Avete gli elementi che vi servono per sapere quanti accordatori ci sono a Chicago, perché voi potete ragionare così.
Sappiamo quante accordature ci saranno praticamente e quante ore serve, cioè abbiamo tutti effettivamente i dati.
Sì, c'è ancora un ragionamento che bisogna fare. Perché tu dici, beh, possiamo abbastanza facilmente calcolare
quanti pianoforti ci stanno, quanti vengono
quante ore ci vogliono per accordarli tutti.
Visto ne viene accordato uno, sappiamo in un anno quanto lavora un accordatore, dividiamo fondamentalmente le ore di lavoro, cioè le ore richieste totali
Sì.
da tutti i pianoforti per le ore di un lavoro di un accordatore, avremo gli accordatori.
Esatto. È esattamente questo. Le accordature per anno sono 9 milioni, fratto due persone a famiglia, quindi 4 milioni e mezzo di famiglie, diviso 20, solo una su 20 ha un pianoforte, quindi 225.000 accordature. Per fare un accordatore può, quante ore lavora? No. 8 ore al giorno, 2 ore per ogni accordatura, quindi può fare 4 accordature al giorno, per 5 giorni a settimana, per 50 settimane può fare 1000 accordature. Ne
Quindi
servono
225.000.
225.000. Supponiamo che l'accordatore lavori più o meno a tempo pieno, altrimenti non guadagna abbastanza da vivere, e quindi a Chicago ci sono circa 225 accordature.
Approssimativamente.
Approssimativamente. Ora, perché funziona questo tipo di approccio? Intanto bisogna avere delle stime ragionevoli delle variabili rilevanti. Il punto è che utilizzando queste stime qualche volta errerete in eccesso, qualche volta in difetto, e questi errori si elidono null'altro, quindi se fate le cose per bene la stima finale è ragionevolmente vicina alla realtà. In questo caso la realtà somiglia a questa. Se voi vedete a questo sito Wolfram Alpha, che è in grado di darvi stime quantitative di un sacco di cose diverse, non trovate proprio gli accordatori di pianoforte, ma trovate il numero di persone che riparano e accordano strumenti musicali in generale. E vedete, sono 350 a Chicago, che è una quantità che si accorda abbastanza bene con quella che avevamo stimato. Ma il punto fondamentale del processo non è raccogliere, questa è solo una dimostrazione pratica, non è raccogliere questi dati, che appunto ce li abbiamo in testa se ci pensiamo. Il punto è trovare una strategia per affrontare la domanda. Se voi riuscite a immaginare cosa vi serve per rispondere alla domanda, poi i dati li trovate. Se non ce li avete, chiedete a me. Bene, adesso facciamo un esercizio. Chi si vuole prestare per primo? Tanto a turno lo farete tutti. Cominci tu perché non ci sarai la prossima lezione. Ah, come preferisci? Vuoi venire qui?
No, sto bene.
Ti sei alzato subito.
No, pensavo dovessi parlare alla lavagna o... Ok.
No, questo è abbastanza facile per cominciare, che si può fare.
All'inizio io ho cambiato.
Quante persone stanno usando un telefono cellulare nel mondo in questo momento?
Ok, devo pensare.
Certo.
A quanti siamo al mondo?
Guarda, la notizia è fresca di oggi.
Sì, eh?
Sì.
Ok.
Secondo le migliori stime, oggi siamo diventati quanti?
8 miliardi e...
8 miliardi.
Ah, spaccati?
Spaccati.
Ok, perfetto.
Pensavo un po' di più, non pensavo più.
No, per fortuna stiamo
no,
rallentando.
Perché
basta,
sentivo tipo 10 miliardi entro cioè saremo così, e quindi ho detto adesso...
Sembrerebbe che le cose stiano andando un po' meglio del previsto.
Quindi siamo 8 miliardi.
Sì.
E di questi immagino che il 70% abbia l'età idonea per l'uso di un telefono cellulare.
Tu immagini che ci sia, che circa un 70% delle persone possegga un telefono cellulare.
Esatto. E in questo momento
Sì.
o forse
Questo.
questo momento
Sono le 17:29 se ti interessa.
Rende più difficile, però magari dipende da
devo
qual è
pensare
l'orario
ancora.
della giornata.
no, qual è il fattore che riesce a discriminare l'uso del telefono in questo momento?
Questo è quello che devi pensare tu, e non si tratta di numeri, si tratta di come approcciare il problema. Voi potete aiutare, se avete qualche suggerimento da dargli.
Posso
In
fare-
media quanto uno usa il telefono al giorno?
ma non è, secondo me è relativo per il momento. Cioè magari in questo momento... chi
Vabbè,
lo
però se
usa-
più o meno 3 ore al giorno le persone usano in media il telefono, l'incroci delle cose, però non lo so.
Che dice Valeria? Interessante ciò che ha detto Samuele.
Cioè al momento mi dà un'idea di
Sì,
tempo, no?
sì, no, in
Il
effetti
tempo
forse ci sta.
associato al
Però
giorno.
Possiamo fare
secondo
la
me
stima.
dobbiamo pensare a quante persone che stanno a scuola, lavorando, dormendo.
Che dici, Kenny?
Ok.
Cioè,
In quali parti del
Io
mondo?
chiedo, non ho detto che
In
né
linea
Samuele
in
né
linea
Valeria abbiano detto
teorica.
una cosa
Sì, sì,
giusta.
Sì, ma ripensando forse il suggerimento di Samuele è sensato e anche... No.
Anche quello di Valeria, a
Anche
questo
quello
punto
di
possiamo
Valeria.
eliminare più o meno un bel po' di
Esatto,
mondo.
se
non lo so.
consideriamo che
Però-
dall'altra parte del mondo magari è notte fonda.
Però vabbè, magari con le abitudini ci sta chi ci sta pure di notte, non lo so, se vediamo... Quante persone, quanto tempo passi al giorno al telefono e quindi manca un...
Quante persone hanno più di un telefono?
Beh, tanto non stanno così di solito.
Ma magari
Voglio che sia
in
un
chiamata e poi con l'altro lo scroll.
No, io sto cercando un modo per passare da ora a telefoni accesi, cioè perché noi abbiamo, che ne so, dal dato, che ne so, le persone usano il telefono in media 2 ore al giorno, ok, quindi noi abbiamo un 12esimo possibilità che in questo momento uno sta al telefono, se c'ha il telefono.
Stai seguendo?
Sì, sì.
Quindi teoricamente noi dobbiamo prendere tutte le persone, abbiamo detto il 70% hanno il coso, un 12esimo di possibilità che stai al telefono, quindi è 8 miliardi.
Un 12esimo al 70% di
Esatto, cioè fondamentalmente...
Concordi?
Sì, più o meno, sì, ora non so...
Non so se
cioè,
ci
non
stanno
saprei
altre
fare
variabili.
una stima sull'uso medio del cellulare.
Vi
perché-
faccio presente, io non faccio lezione sempre alla stessa ora, però la diapositiva non la cambio. Questo ragionamento è valido sempre, non importa sapere se i cinesi o i sudamericani sono al buio o sono di giorno in questo momento. Mediamente, stiamo parlando di stime, più o meno tante persone dormiranno e tante saranno sveglie.
È il motivo per cui funzionava, esatto, è una cosa che si
E quindi il
bilancia.
procedimento è esattamente, era un
Vabbè.
paio d'anni fa questa diapositiva, ma ci abbiamo messo 12 anni, passare da 7 a 8 miliardi. La popolazione mondiale, usiamo questi numeri, non devo stare più neleggiare, è di circa 7,5 miliardi di persone. Supponiamo che 2/3, adesso un po' di più, 70% va benissimo, usino un cellulare, diciamo 5 miliardi di persone. Io possiedo un telefono cellulare, dice Samuele, e lo uso circa 3
Io ho
ore al
detto
giorno.
2.
Alcuni studenti dicono 5 ore al giorno tranquillamente, io rimango veramente scosso, ma se è così, diciamo 3 ore al giorno. Ritengo che il mio uso del cellulare sia abbastanza prossimo alla media mondiale, oppure uno può dire no, invece io lo uso un po' troppo, gli altri in media
Secondo me.
stimate. Di nuovo sono stime, sono approssimazioni. E quindi i 5 miliardi di persone che usano un cellulare lo fanno per 3/24esimi della giornata, quindi 3/24esimi di 5 miliardi fa 625 milioni. E questa è la risposta. Circa 625 milioni di persone stanno usando il cellulare in questo momento. Quindi
È preoccupante un po'.
Beh, diciamo, mentre quello degli accordatori di pianoforte può essere interessante anche quello, sì, se voi volete sapere se è una buona idea andare a Chicago per fare l'accordatore di pianoforte o no, quanta concorrenza avrete, può essere un dato interessante. Se siete un ingegnere aerospaziale volete sapere grosso modo quanto deve essere grande la banda dei vostri satelliti per connettere i cellulari di tutto il mondo, vi può interessare prima di fare tanti calcoli avere un'idea approssimativa. E Questi adesso sono esempi un po' scolastici, ma poi passeremo ad esempi reali, concreti, di laboratorio, vi farò vedere a cosa servono questi esercizi. Ora, in realtà siete stati ben più bravi della media, quindi passiamo a un'altra domanda che non avevo intenzione di fare oggi, supponevo avremmo affrontato la prossima settimana, ma la possiamo fare. Allora, ecco la prossima domanda. Oh, no, prima di farvela vedere voglio un volontario. Karim ha già risposto, uno di voi due.
Valeria.
Lei?
Non è volontario quindi.
Ci stai? Va bene.
Tanto
Allora,
mi aiuta
Quanta-
molto.
Come con Karim, poi magari ragioniamo assieme.
sì, sì, beh, perché come dicevamo prima, no? È
Eh,
così
una
che si
collaborazione.
elaborano le
Brainstorm.
cose.
Cascate
Bene,
delle gare. No.
qualche anno fa erano le cascate di Niagara, sì.
Sembrava.
Ci abbiamo già azzeccato, prossima domanda.
No.
Non è così.
Quanta acqua scorre? No.
Questo è il versante americano, io in realtà stavo su quello canadese più o meno qua. E a un certo punto il fratello di mia moglie, che non per nulla è fisico, mi dice: secondo te qual è la portata delle cascate del Niagara? Io ho detto: adesso te lo dico. Mi sono affacciato, ho dato una buona occhiata intorno, ci ho pensato un po' sopra e gli ho dato una cifra. Che ho fatto?
La definizione di portata è...
Metri cubi al secondo.
Ok.
Quantità d'acqua che passa per
passa,
un determinato
sì.
piano che tu metti.
Volume nel tempo.
Sì. Allora, prima di tutto ci vuole un approccio. No, io vedevo la situazione reale, quindi non ti chiedo di fare stime su questa immagine che non puoi valutare, non hai. Quindi poi i dati stimati te li do io, ma tu mi devi dire che cosa ti serve. La situazione non è molto diversa da quella di prima. Prima abbiamo
No.
drasticamente semplificato il problema. Abbiamo detto sì, va bene, non importa chi è al buio, chi è sveglio, abbiamo diviso grossolanamente la popolazione per un certo numero di ore, abbiamo detto questa. Ecco, qui che cosa possiamo fare? Qui stiamo parlando di volumi, invece, volumi al secondo. Volumi per unità di tempo. Allora, le cascate sono tutta questa roba qui. Qui dietro c'è un pezzetto che si vede, e qui finiscono. Valeria non ha un'illuminazione, qualcuno vuole aiutare?
Penso sicuramente la misura, la lunghezza della cascata.
La lunghezza delle cascate.
L'altezza.
Questa è una stima.
L'altezza secondo mi domandavo se fosse relativa o se...
Loro hanno detto lunghezza, altezza. Perché vogliono questi dati? Perché hanno qualcosa in mente. Cosa hanno in mente?
Altezza non so se è rilevante.
Andiamoci alla lunghezza. Che ci
Lunghezza
fa?
perché cioè se è più estesa è facilitato il passaggio di acqua.
La portata di un fiume è la stessa in ogni suo punto, perché se l'acqua passa da un punto deve passare da un altro nello stesso modo. In effetti il fiume si attraversa dove è più largo, perché la portata è distribuita,
Ah, ok.
è meno profondo dove è più largo. E non come fece un mio amico che mi disse: ma passiamo di qui che è più stretto. Blocco.
Io.
Quello che
Facciamo prima.
Abbiamo detto che stiamo cercando un volume al secondo, non è così difficile.
Non ci serve intanto una stima quanta acqua...
Beh, quella è quasi la risposta finale.
Ok.
La velocità con la quale scorre,
Eh?
la velocità con la quale scorre
Eh.
è importante, immagino.
Sì, perché
Eh.
la risposta implica un fattore tempo, quindi da
Quindi.
qualche parte il tempo che dobbiamo dare fuori.
Beh, allora se abbiamo una stima della velocità, poi
Molti
cioè la forza di gravità non so.
Io non ho detto che abbiamo una stima della
Ah.
velocità. Perché io perlomeno non sono capace di guardare il fiume.
Infatti.
E allora ci serve l'altezza.
L'altezza io ho pensato perché, vabbè, la forza di gravità comunque è quella che spinge giù l'acqua e quindi c'è il tempo che ci metterà, cioè non lo so,
Sì.
cioè mgt è uguale a un mezzo mv quadro alla base, non lo
Sì, ma
so.
è meno complicato di così, non
C'è
serve.
la conservazione dell'energia, che ne so. Però per
È un
stimarlo.
problema gestibile a mente. Abbiamo la lunghezza, mi pare che siamo d'accordo anche tu che la lunghezza in qualche modo ci serve.
Sì, almeno è un dato che potremmo
Beh, allora la
stimare.
mia stima era che la lunghezza attuale delle cascate fosse 1 chilometro. Stima di
Ok.
ho guardato e ho detto, ma
Sarà più o meno.
quindi era ovviamente possibile.
Sono alte, una trentina di metri di più.
Quindi ci interessa l'altezza perché qualcuno invece era in dubbio. Ci
Io ero in
interessa, non
dubbio.
ci... Tu eri in dubbio.
Ero in dubbio.
Hai cambiato idea.
Ho cambiato idea, ora è più idea.
No, forse non ci sono io prima.
Tu hai cambiato idea?
No, vabbè, troppo cervellotica come cosa, effettivamente.
Cioè se non cambierebbe qualcosa se fosse alta 30 o 40 metri.
Non lo so.
Non lo so.
No, a me era solo per
Non
ricavare
lo sai,
un dato.
lo sai dire perché non hai ancora trovato come approcciare il problema. Ti si chiede un volume, lascia perdere i secondi per un momento. Come l'approcci il problema del volume?
Il volume è area di base per altezza, per esempio, se fosse...
Quindi quante dimensioni ti servono per calcolare un volume?
Sì, cioè mi serve anche l'altezza,
E
un'area, quindi
la quale a sua volta un'area come la calcoli?
se io ho una lunghezza mi servirà anche una larghezza in qualche modo
Sì.
e l'altezza chiaramente,
Sì,
e sono 3 dimensioni,
sì.
non so come...
A che somiglia, che cosa semplice, a che oggetto semplice somiglia una cosa che ha una lunghezza, una larghezza e una profondità?
un cilindro?
Rettangolo.
Parallelepipedo?
Più sì, una scatola diciamo, come se fosse una scatola.
Allora,
Ma allora a noi ci serve però la profondità più che l'altezza. Cioè, noi il volume è quello d'acqua quello che ci interessa, non quello della cascata. Quindi, Enrico,
No, penso intenda l'altezza in cima. Se intendi cioè il livello proprio dell'acqua prima della cascata, no?
sì, non ci
Per
serve.
sapere quanta poi
ci potrebbe
effettivamente ne
essere utile,
passa.
certo, ma tu sai dire che profondità
no.
l'acqua qui, io no.
E poi dall'altro punto soprattutto di questa cosa. Non è detto che sia omogeneo, no?
Non è detto che sia omogeneo, però mentre è difficile dire che profondità ha l'acqua qui, si può fare una stima di quanto è alta la cascata.
Quello.
Questo esercizio è molto più facile su una cascata che non guardando un fiume che scorre e non ho idea di quanto sia profondo.
Quindi ci
No,
serve.
ancora non c'è stata l'illuminazione. Devi cercare il volume di quest'acqua sostanzialmente.
Quella che passa.
Come si fa? Come si fa il volume di un parallelepipedo?
Ma non vedo il parallelepipedo.
Non lo vedi? linearizza questa cascata, invece di seguire tutte queste curve,
Sì.
fanne una linea retta.
Sì.
Lo vedi il parallelepipedo adesso?
Ah,
Hai una larghezza o lunghezza,
Ok.
chiamala come vuoi.
Sì.
Sì.
Le altre due dimensioni?
L'altezza.
Eh?
L'altezza.
Il preso pensa al tempo.
Ok.
E poi
E poi? Qual è la terza dimensione?
è in base al peso specifico, penso, dell'acqua per...
Cioè,
Vedi
dobbiamo
una cosa per
fare-
volta, stiamo arrivando alle dimensioni. Allora abbiamo
Cioè,
una
dovevo
quella
vedere
che-
quanto si estende.
Vabbè.
Cioè, da dove arriva?
Come chiami la lunghezza complessiva delle cascate? Lo chiami larghezza? Voglio usare le tue parole.
Ok, lunghezza.
Lunghezza, benissimo.
Poi abbiamo l'altezza.
Benissimo.
E
Che
poi
altro
mi
ti
serve
serve?
l'estensione, cioè la larghezza.
La larghezza, cioè
Non lo so.
se questa è la lunghezza e questa è l'altezza, dove sta la larghezza? È un parallelepipedo, abbiamo detto.
Cioè, sì, è per...
Per forza.
Ma di da dove?
Che cos'è qui la terza dimensione?
Cioè...
Così la vedi la terza dimensione?
Ah, ok, quindi cioè quanto l'acqua si discosta quando cade.
La profondità della cascata.
Sì, ok.
Questo è il parallelepipedo di cui stavamo parlando in sezione. Ok? Allora, immaginiamo, come ho fatto, che io abbia stimato la lunghezza, l'altezza e la profondità.
Però la profondità, esatto, cioè è difficile da stimare. Io pure prima cioè,
Sono andato in un certo posto in cui si vedevano le cascate quasi di profilo,
ok.
c'era una particolare finestra in cui si vedevano e ho fatto una stima. Qualcosa del genere, vediamo che cosa... sì, ok. Allora, ups, no.
Troppo.
Ok, abbiamo il volume della cascata.
vabbè, sì, ho letto, cioè sapevo che mancasse il
Zitto,
tempo.
se hai letto stai zitto.
No, però ho comunque domande perché ho letto rapidamente, ho visto solo la
E allora
soluzione.
ascolta.
Quindi, ma abbiamo quindi questi tre valori, altezza...
Diciamo che abbiamo il volume della cascata.
Sì. E poi...
Però abbiamo detto che dobbiamo fare
il
la-
tempo, quindi...
la portata del spino in metri cubi al secondo.
Sì. Quindi quanto ci mette l'acqua da arrivare da in cima a fondo? E quindi...
Lo possiamo stimare?
Questa era la mia domanda, lo possiamo stimare questa
cioè, non
cosa?
è
Sì,
la
la guardi.
caduta di
La
qualsiasi
guardi è
cosa.
facile,
Scusa?
Fissi...
Cioè-
la segui.
Ah, segui.
Che in funzione dell'altezza qualsiasi oggetto cade allo stesso tempo,
Sì.
in teoria.
Come disse già Galileo.
Ah, ok, giustamente, non
Cioè
la
la
possiamo
massa non è
guardare.
rilevante per un oggetto, ok. Quindi l'accelerazione di gravità no, non ci interessa.
Faticose da calcolare a mente accelerazione con i quadrati, mamma mia.
Beh, se abbiamo l'altezza allora lo possiamo effettivamente
Stimare.
stimare a me, cioè,
Ecco.
beh, se è alto 40 metri l'accelerazione di gravità effettivamente la assumiamo come 10.
Faccio una cosa, il cronometro.
Che da Google.
Supponiamo che ci sia un ramo che passa in questo momento, io guardo
Devi andare tipo a fermare.
quello che ho fatto nella realtà era prendere un punto cospicuo, ecco, un particolare spruzzo e vedere quanto ci impiegava questo spruzzo a arrivare sul fondo.
Abbiamo tutto.
A questo punto abbiamo tutto, perché abbiamo detto, abbiamo capito che
Il volume.
il volume è un parallelepipedo ideale che ha le dimensioni della cascata. abbiamo delle stime di queste dimensioni. Abbiamo stimato che la lunghezza è 1000 metri, che l'altezza è 20 metri, queste sono le mie stime, che il tempo di caduta, che la profondità è 2 metri e il tempo di caduta è 3 secondi. Questo ci dà 1000 per 20 per 2,
Diviso
40.000 diviso 3 secondi, 13.000 metri cubi al secondo. Bene, qualche minuto dopo, continuando a camminare, ci imbattiamo in un cartello con i dati
Ah,
già pronti e sciorinati.
stavano cercando di...
E quindi vediamo i dati reali. Allora, lunghezza 1110 metri, leggermente sottostimata. Altezza 45 metri, sensibilmente sottostimata. Profondità non era data.
Perché irregolare?
Eh?
Perché irregolare la parete o...
Perché irregolare, perché la profondità della cascata è un concetto piuttosto vago, lo puoi usare come stima ma non è che la puoi misurare realmente. Il tempo di caduta è effettivamente 3 secondi. Allora qui c'erano 2 stime in difetto e una in eccesso. Vedete la tendenza a compensarsi dell'errore e vedete il risultato che dava il cartello, 3340 metri cubi al secondo. Ora, voi dite, beh, ma hai sbagliato di parecchio, dipende. L'ordine di grandezza è corretto, cioè parliamo di parecchie migliaia di metri cubi al secondo. Quindi diciamo come stima approssimativa non è male, nel senso che se torniamo per un attimo, ecco, questo edificio che a malapena si intravede qui dietro è una centrale elettrica. Se io ho bisogno di sapere se vale la pena o no di installare una centrale idroelettrica su questo fiume, prima di cominciare a fare calcoli complicati dovrei avere un'idea della portata, cosa che posso misurare, non ho bisogno di un igrometro, ho bisogno di una serie di misurazioni eccetera, e invece posso fare una stima di questo genere. Quindi l'ordine di grandezza era già indovinato, di più presumo che il cartello avesse qualche sorta di errore, perché se voi sostituite
ma infatti io stavo
qui viene una
vedendo.
profondità di 20 cm, ora che la cascata abbia questo spessore, no, non funziona. È probabile che in realtà la portata reale fosse maggiore di questa, quindi questa stima era anche migliore del reale. In più la portata delle cascate del Niagara varia moltissimo fra l'estate e l'inverno perché dipende dallo scioglimento dei ghiacciai e quindi non so esattamente queste misure a cosa si riferissero, però diciamo non è male. A priori non avevo la più pallida idea, se mi avessero detto 1 chilometro cubo al secondo avrei detto bah sì, forse è possibile, non so, ma vedete con un ragionamento non particolarmente difficile sono riuscito ad arrivare a una stima ragionevole. Il punto era proprio quello di visualizzare un approccio al problema, ridurre una cosa complessa a qualcosa di approcciabile, soprattutto approcciabile mentalmente. Parlo delle vite.